Howdy folks, in this microbiology video we're going to be talking about how to do the quad streak. Now the quad streak is a method that we use with plates in order to isolate out individual colonies from a culture of bacteria. Now, we're going to draw this at first here on the board just to help you visualize what will be harder to see in this video on a plate. With a quad streak, here's your representation of your petri dish. This is where you're going to have media within that you're going to brush your bacterial culture across. Your first time of inoculating this plate for the purposes of a quad streak, you are going to draw bacteria from your bacterial broth in a tube in your inoculation loop. Close that bacterial tube, put it aside, and never come back to it. Okay? With that inoculation loop now, you're going to take the tip of your inoculation loop, which is illustrated by the marker here, take the tip of the inoculation loop and simply brush it back and forth near the edge of the plate. Okay? In this process, you have a lot of bacteria that will grow up here near the beginning of your brush stroke, and it will begin to taper off the further down in the brush stroke you get. Alright? Now that's just your first inoculation. Once you do that brush, you want to take your inoculation loop, sterilize it, and come back. Now do not return to your tube at this point. The idea is to isolate individual bacterial colonies, and if you go back to your bacterial culture broth, you're just going to keep adding more and more bacteria to the picture and not be able to isolate anything. So with your second pass with the inoculation loop, once it's been sterilized again, you're going to start outside of the first inoculation brush path. Start outside so that you don't risk potentially burning the bacteria if you didn't wait long enough with your inoculation loop. Start outside so you don't burn. Bring your brush strokes into the path of the first, just barely, at maximum twice, and then continue to go back and forth. Again, spreading out those few colonies that you happen to, those few bacterial colonies that you happen to pick up there with that inoculation brushing. Again, sterilize your inoculation loop and come back for a third inoculation. This is called a quad streak, so we're going to do 4 passes. And you simply do this again. Once your inoculation loop is sterile, again, do not return to your bacterial culture tube. Start outside of the second inoculation brush path. Sweep in, just barely. Sweep in, and then stay out. Alright? Sterilize your inoculation loop, and come back for a 4th pass. Quad streak, you're going to make 4 passes. At this point, again, with a sterile inoculation loop, you're going to start outside, sweep in just barely, and then you are going to potentially cover the rest of the plate with brush paths. The reason for this is, in our first path, our green path, we have a lot of bacteria that are going to grow because that's where you started, that's where you seeded the bacterial culture. Then you pulled a few of the bacteria out that will then grow more cultures here, but less than what we'll see in the green path. You'll pull some of those cultures out in the orange path, and you will have some growth here, but less than what you see in the red path. And then finally, with this final path, you will see only a few colonies show up if you've done everything accordingly. So, now that we've illustrated how to do a quad streak, let's show you how to do it with an actual plate. So here's that plate. Again, you're going to want to make sure your inoculation loop is sterile, so we need to turn on your Bunsen burner. Okay. Make sure you got a good cone of flame. All right. Grab your inoculation loop, sterilize it. Again, make sure the stem is red hot. Work your way up to the loop itself and let it cool off next to the flame so that it doesn't risk picking up any airborne bacteria while you're waiting for it to cool down. Now here's our culture broth. This is where we have grown some bacteria so that we can then isolate individual colonies on this plate. So as we go in to pull from this tube, again, hold it sideways, get your inoculation loop between your fingers so that it's steady, grab the cap with your pinky, remove the cap, hold it at an angle to reduce potential contamination, flame the mouth of the tube, input your inoculation loop into the liquid, collect a little bit of liquid, swirl it around some, remove the inoculation loop, flame the tube, and cap. If you have a little bit of bacteria drip, like we just had there, you're going to want to make sure that you come back with isopropyl alcohol to sterilize the bench top. But for right now, let's go ahead and get this inoculated. So you're going to take your plate, lift up the top just enough so you can get your inoculation loop in, and very lightly brush back and forth. And as you do this, you will see some slight markings on the media where your inoculation loop comes in contact. You need the right angle and light contrast to see that. It's not terribly hard to see, and with time and practice you will be able to see it just fine without any issues. Sterilize your loop once more. Do not go back to your original bacterial culture. Set it aside. You're done with that. For your second pass, you generally have an idea of where you stopped brushing, so look in that region of the plate to find those sweep marks. Start just outside of that path. Bring it in, out, and brush back and forth outside of that first path. Cover, sterilize, wait for it to cool down. Go ahead and turn your plate so that it is in a position that is easiest for you to then make your next brush path. Once it's cooled down, bring your inoculation loop in. Start outside that last path. Just barely touch, sweep in, and stay out. Close your dish. Sterilize your loop. And wait for it to cool down. Go ahead and turn your plate one last time for our fourth streak. Thus, this technique is called the quad streak, colony isolation. Once it's cooled down, lift your plate one more time. Start outside the previous path, bring it in, bring it out, bring it in, and stay out. And then you can bring that all the way across everywhere where you see you have not touched with the inoculation loop yet. Never pick up and go back where you have been. If you do that, let's not forget our last sterilization, if you do that, you're going to lose your potential isolation. Okay? So, that is our quad streak video. And don't forget, we need to sterilize that spot where we drop some bacteria, and we'll do that.